Thank you, Eduardo, for the presentation. As Eduardo mentioned, I am postdoc at Costa's lab, and today I'm going to present you my latest research work that we have entitled "Differences in Metagenome Coverage: My Confound Abundance Base and Diversity Conclusions." And well, this is the index of the presentation that I'm gonna give you today. Along this presentation, I will try to answer these 4 questions. First of all, why should we avoid comparisons between metagenomes with different non-parallel coverage? What should we do instead? In which situation we should worry about differences in non-parallel coverage, and what is the best normalization approach depending on our aims? So, well, for those who are not familiar with non-parallel coverage, this is a metric that was proposed back in 2013 by Luis Miguel Rodriguez and Kostas Konstantinidis. And basically, this metric, the non-parallel coverage, measures the fraction of the total DNA diversity that is sequenced in a metagenome. So this is a metric that measures how representative our metagenome is from the actual diversity in our sample. Based on this tool is based on read redundancy, and as you can see here, when we plot the non-parallel coverage, the metagenome coverage here on the y-axis versus the sequencing effort on the x-axis, you can see that at very low sequencing effort, the coverage is very low. But as we increase our sequencing effort, the non-parallel coverage increases until we reach a plateau, meaning that we have reached a complete metagenome, a very good representative metagenome. However, as you can see, the trend is different for different kind of samples. For example, for human microbiome, the sequencing effort that we need to apply to reach a very high metagenome coverage, it's very low. It's 0.1 gigabyte. However, when we are analyzing environmental samples and specifically soil samples, the sequencing effort that we need to apply to reach the plateau is much higher. For example, in this case, 100 gigabytes or more. One important thing that I would like to highlight that metagenome coverage is a different metric that should not be confused with depth of coverage and breadth of coverage, that are 2 common metrics frequently used in read recruitment analysis, but measure different things. So please, these are 3 different metrics that should not be confused. So what's the point of our work here? So in this plot, I show you the number of manuscripts on the y-axis that were published over the last decade on the x-axis that have used metagenomics in their analysis. And as you can see, they have exponentially increased. However, the number of these manuscripts that have also considered non-parallel in their analysis is still very, very low. Indeed, only 0.5% of all studies using metagenomics have considered non-parallel in their analysis. And this is worrying, and I will show you why. So here on the panel on the top, I show you the aggregated relative abundance of a group of MACs belonging to the phylum Pseudomonadota and the phylum Thermoproteota at different non-parallel coverage, ranging from 0.1 to 0.7. So as you can see here, the relative abundance of the aggregated relative abundance of the Pseudomonadota increases as non-parallel coverage increases. However, in the case of the Thermoproteota, this relative abundance remains more or less stable. But what is the reason behind this difference? So on the second panel here, I show you on the y-axis the number of MACs detected for each of these 2 phylum, again as a function of non-parallel coverage. And as you can see, for the Pseudomonadota, there is a continuous increase in the number of MACs detected as our non-parallel coverage increases. Something that does not happen with the Thermoproteota, that has more or less the same number of MACs. So basically, the differences in the relative abundance that we observe as non-parallel coverage increases is due to the different sensitivity of the metagenome. So basically, as we sequence more, as we increase non-parallel coverage, we are able to detect more MACs belonging to the pseudomonadota as opposed to the thermoproteota. And I can say that this is not due to an increase in the relative abundance of individual MACs, because this is specifically what I'm showing you here in the third and fourth panel for the pseudomonadota and the thermoproteota. Each line here represents one MAC at different non-parallel coverage, and as you can see, as far as we are able to detect the MAC, the relative abundance it's reliable, it's stable, and we can use it to compare between different samples. So I hope I can convince you that if we are comparing different metagenomes, and if we are comparing groups of features, let's say MACs or genes, if we are grouping these MACs or genes into a category, we should worry about differences in non-parallel coverage. Going to the second question that I have in the index, what should we do instead? Well, the answer is simple: normalize to the same non-parallel coverage, and I will show you why. Here on the x-axis, I showed you the relative abundance of a group of MACs belonging to the marinosomatales in a vertical profile on the ocean. The solid lines here show you the data with the full original metagenome, and as you can see, this group of MACs peak at 150 meters. This is the same trend that I observe when I compare metagenomes that were normalized to the same non-parallel coverage, in this case 0.5. As you can see, the values are different because of the different sensitivity that I showed you in the previous slide, but the trend remains the same. However, this is not the case when we compare metagenomes with an uneven coverage. As you can see here, what we got, it's actually a drop instead of a peak in the abundance of the marinosomatales. So if we were analyzing metagenomes with an uneven coverage, then the conclusion that we could get from this data would be wrong. And probably some of you might think that this kind of errors are not published in the scientific literature, but I can tell you that they are. We reanalyzed this manuscript where there were differences in non-parallel coverage between different types of metagenomes, and we observed that the antibiotic-resistant gene richness, the diversity of antibiotic-resistant genes in effluent, was overestimated due to differences in non-parallel coverage compared to influent and sludge samples. And something similar was observed with the differential abundance analysis that the authors did. As for the third question, when should we worry about this error, about this bias? Well, the answer is also simple. The lower the abundance the feature you are interested in, the more likely the bias. And this is true for different environments. As I show you here, the analysis we did for seawater, human gut, freshwater, and pit soil metagenome. And as you can see, the lower the relative abundance, also the lower the maximum the difference in non-parallel coverage that we can accept to get unbiased result. So if you are working with low relative abundance features, you should worry about this bias. Finally, as for the fourth question, what is the best approach to normalize our data? We have provided a decision tree depending on your aim, and we have also provided some scripts to facilitate the normalization step. So as for regarding conclusions, I would like to say that, as I was saying, metagenomes with different non-parallel coverage should not be compared due to the different sensitivity. Instead, we should compare metagenomes with an uneven coverage, especially if we are analyzing features with a very low relative abundance. To facilitate the normalization, we have provided some scripts that you can find if you scan this QR code, and also a decision tree. And finally, no matter the conclusions you got, you should always restrict them to the non-parallel coverage you have used in your analysis, because if you are able to sequence more, your results could be different. So unless you get a very complete metagenome, you should always restrict your conclusions to the non-parallel coverage you use. So, well, thank you all for your attention. Thank you, of course, to my postdoc supervisor, Kostas Konstantinidis, and our collaborator, Luis Miguel Rodriguez, to our sponsor, to MVIF, for the organization of this nice event. And, well, these are my contact details in case you want to reach out to discuss about this paper or for future research opportunities. Thank you.